Top, it's 5 o'clock. My name is Florian Eigenmann, and I'm giving you an introduction on QCL frequency comm, specifically the benefits and some selected applications. And then later, Carsten Kötting from the Garrett Group in Bochum will give the body of this talk and what we measured last year during the first or just before the first lockdown in Germany. Quick update or just the main benefits of dual comm spectroscopy. So the benefits here, the main benefits are speed, brightness, broadness, and especially compared to single laser spectroscopy to analyze complex mixtures, to determine, for example, secondary structures in amide 1 region. And then last but not least, high resolution. Some data I want to present here. That was already published early on with bacterial rhodopsin measurements. And this was published in 2018. It was validated with StepScan FDIR for repetitive reactions. Together with Tilman Kotke and Jerome Feist. The other advantage or other benefit is the brightness due to lasers. So we get actually 2 or 3 orders of magnitude stronger lasers, brightness, laser power with the laser which is stronger than glow bar source, which allows single shot analysis and gives an excellent SNR in a short amount of time. By sweeping the comms, it's also possible to do high resolution spectroscopy, and we recorded already better resolution than 0.001. This is actually highlighted, or it's we go into details with some other webinars, which I will give a brief overview later on as well. Then the other benefit is the parallel multi measurements, also that we are not just recording one single laser line, so we can measure over 60 to 100 wave numbers, and this is shown here in a heterodyne signal, which you can see here the results of a water vapor spectra. This is done here from a hydron water vapor spectra, and those lines match up very nicely in the 1650 regions. The 2 frequency comms, I will not go into details, but there are 2 comms, laser comms, which are mixed on a beam splitter and detected on a high bandwidth MCD detector. So each pair of those optical modes produce a radio frequency beat node, and the radio frequency spectrum can be measured with conventional electronics and represents the optical spectrum. So why is that so important, and why is that so fast compared to other spectroscopic techniques, for example, FDIR? So there are no moving parts, so you can really measure single shots and fast microseconds, as Carsten will show later on. A big advantage of the IRsweep of this spectrometer is that you can change the laser within a few seconds. So there's actually a movie here where Pierre, one of our senior scientists, engineers, change such a laser module. He disconnects it here from a chiller, so those laser modules are stabilized, temperature stabilized very accurately. And then you can change such a laser module and put it in 30 seconds. Stabilize it, of course, a little bit longer, but probably after 5-6 minutes, you can do very accurate measurements. Every time we present new lasers, and I'm very proud actually that we fill the gap as we speak, and we have already new lasers available. I had a talk to this AM with Andreas Hugi. So we have at 1490 an additional laser, which is very exciting that we can also put that on the list. It's really fresh. And I will highlight another one. That's the 1240 laser, because that's one of the lasers we did most of this data, or we recorded most of the data which Carsten will present later on. And 1650 was also used during this study. Some applications which you will find as well on the page is definitely about protein dynamics, and it's also like we have a lot of customers actually. They are doing such webinars specifically during the lockdown. We had a webinar with Joachim Hipperle on protein monitoring. That's definitely also a very interesting part, which might be relevant for most of you. Then a webinar with Nico Pankowski, hosted on fast measurements on at a shock tube application on high temperatures. Then a webinar with Ted King about stop flow applications, and then with Ian Burgess, we also did the speed advantage, highlighted the speed advantage of the IRsweep. Then we had two high-resolution spectroscopy webinars, one with Muriel Lepere about gas mixture studies, and one with Lukas Ebenecke, more on the technical side on high-resolution gapless dual-com spectroscopy with current-tuned QCLs. And then last but not least, a very exciting webinar, which we had end of last year with Jérôme Feist about the latest development on QCL frequency comms. All these webinars you will find directly on the page. It's a little bit differently organized, but I just highlight that here as well before I go over some selected applications. Selected applications I want to highlight today before we go to protein dynamics is one is stop flow, and the other one is some mid-IR PAT, process analytical technologies, and fiber optics coupling with 10-meter fiber ATR probes. So first, stop flow, it's very exciting actually. We had almost a year now, just before the lockdown, we received such an equipment from Ted King, and we measured a lot of data during the lockdown, specifically because mid-IR is very interesting, and such fast measurements is much more specific than UV vis and fluorescence. And up to today, there are no mid-IR data possible in microseconds or millisecond time regime. So the first results we received here, this is actually also covered in a specific webinar, but I just want to highlight it's a kind of a test reaction of this methyl chloroacetate with some potassium hydroxide. We increased a little bit the concentration to speed up the reaction, and we got really nice results, specifically here in the order of a few milliseconds, and we were able to determine a lifetime of 47 milliseconds. Another result with a very good signal-to-noise ratio was the ubiquitin folding, where we also monitored the clear evidence. So we had we observed the clear evidence of beta sheet conversion with a lifetime of 1.5 seconds. We are not working actually exclusively with Ted King, so we had also from Biologique, Cédric Georges here in Zurich. He came also, I think I was just recently on the phone with him. He was, that was the only trip from France to Switzerland he did last year. And we were also able to measure some really nice data, which is covered in an application note, and he just confirmed recently that he will also do a webinar probably in May. Results outlined here, it's a denaturation of beta-lactoglobulin by 3-fluoroethanol, and you really nicely can see an additional band here of some data where we see even an intermediate. We are very proud actually that this is now possible with dual-com spectroscopy. Then another application which is very promising is the mid-IR fiber coupling, which you can see here. So this fiber coupling work which was done together with Adrian Daly from Pfizer. So those measurements were also performed end of last year. It's a fiber which we got from Art Photonics, 10-meter ATR fiber, and this shows really nice result in the area at 1200. We were also measuring with a 1240 laser, and we measured some acetone in toluene. That's a reaction which a lot of leading FTIR companies do just to measure the performance of an ATR fiber. And the big innovation here is definitely that we had such a fiber up to 10 meters, and those data were recorded in less than 250 milliseconds, and with the current processing time, less than a minute, we got those data points. So this is very exciting news. We just presented that. I presented that at IFPAC. It's a process analytical show in the US. Of course, these days it's everything virtual, and the feedback is already great. Last but not least, and I want to go now to protein dynamics, where we did a lot of work. As I mentioned before, 2018, those results were already published about the cyclic measurements with dual-com spectroscopy. Then we had a lot of work and some additional webinars, which was a webinar which was recorded from Joachim last year about some reactions, specifically cyclic reactions, but he did already also these non-cyclic reactions, which will also be important now for this talk. We have measured just before the lockdown non-repeatable protein reactions with cage compounds in the Garrett group in the nice new building in Bochum at the Prodi. And before I hand over now to Carsten and want us to introduce him. Carsten, are you here already? Hope so. Yeah, it's good to.
Yeah, I'm here.
It's great, actually, great timing, because I was just not sure actually if we are right on time, but okay, I will introduce Carsten. So Carsten Kötting obtained his PhD in the physical organic chemistry department of the Ruhr University in Germany, 1999. He was working on a matrix isolation of carbines, and afterwards Carsten joined the group of Nobel laureate Ahmed Zewail at Caltech, where he did femtochemistry of organic intermediates. After 2 years, he returned to Ruhr University, but this time to the Department of Biology and Biotechnology, and joined the Biophysics Institute of Klaus Gerwert. Carsten finished his habilitation in chemical biology in 2009. He's currently working on time-resolved FTIR spectroscopy of proteins with the main focus on GTPases. Within the new Center of Protein Diagnostics, Prodi, he's developing new chemical methods for the immobilization of proteins to extend the applications of vibrational spectroscopy. So Carsten, please go ahead. You can just share your screen, probably, or just take your slides over. Then you can also use animations.
Yeah, so I think you should now see my screen. So thanks, Florian, for the introduction. So you already heard a lot about the dual-com spectroscopy, about the first line of the title of this talk. So I will now talk about the non-repetitive protein reactions, which type of proteins we are looking at, and also about the cage compounds. What are cage compounds and how can we use them to investigate proteins? Yeah, so most biophysicists, including me, like to work with proteins which do something with light, which have a photocycle, like the rhodopsines. But if you look into your body, most proteins will do nothing if you shine light on them. And obviously, they are also very interesting to look at. And if you want to do that, you can use the cage compounds. And cage compounds are molecules which are somehow caged. They do nothing with the environment, but if you shine light on it, the cage group will be cleaved off, and then you have a biological active molecule. And this molecule can be a phosphate, a carboxylate, it can be a metal, other small molecules, carbon dioxide, electrons or protons. So there are really a lot of choices, and you can investigate lots of protein reactions by the help of cage compounds. What we do is we look at GTPases, like the famous RAS protein that is shown here. And this is a GTPase. So what you need is then the caged GTP. And this is shown now here. You see here, this one, I have to start my pen. Yeah, so this is GTP, and you have here the cage group. And this is already bound within the RAS protein in the binding pocket of the GTP. You can see here the cage group, and by a laser flash, you cleave off the cage group and you end up with a natural GTP, and only then you can investigate the hydrolysis reaction of the GTP. This you have to do with rapid scan, FTIR usually. Obviously, I will come later to the advantages of dual comp, but most of our work was done with FTIR so far, and there we stick with a time resolution of about 10 milliseconds. Yeah, if you have a larger proteins, you have to measure longer. You will have time resolutions about 50 milliseconds, 100 milliseconds, but in principle, the technique can give you 10 milliseconds time resolution if you have 6 wave numbers resolution. Yeah, our sample is just then here in the spectrometer, the protein solution in a buffer with high concentration. And we can irradiate this with light to start our reaction. Yeah. If you want to measure faster, most of you probably know the step scan technique, where you fix your moving mirror in a certain position, then you do a time-resolved experiment, then you move to the second position, and you do the same experiment under the identical conditions in the second position, and so on. And then after maybe 500 positions, you can then slice your time-resolved data into interferograms to have then interferograms with about 10 nanoseconds resolution. However, for this, it's very important to have exactly the same conditions after the reactions for each cycle. So that works very nicely with proteins which have a photocycle. But if you have a cage compound, you flash it and then it's gone. Yeah, there's one workaround which was done by Klaus Gerbert already more than 20 years ago. There the whole sample was divided into small 20 micrometer by 20 micrometer segments. And then by means of an infrared microscope, this was then measured point by point. And by that, you have identical environment everywhere. And if your sample is very homogeneous, you can then do a step scan experiment with this sample. However, to get good data, 5 samples were needed, and the overall measurement time was 5 hours to get, in the end, here the time resolved data from the caged ATP. Yeah, and we measured that in February 1st with the new dual comp technique, and we get very similar half-life time here of the reaction. However, the measurement was done within 1 second instead of 5 hours. And it's really just a one-shot experiment, and we get the data out of it. Yeah, I just told you after the decaging, we have the GTP. Why do we actually have a half-life time here anyway? The reason is that this is a nitrophenyl ethyl cage group, and this forms an intermediate anion, and this has a half-life time of about 50 milliseconds at room temperature. Also depends on the pH. So this is usually fast enough to measure with rapid scan FTIR, but it's clearly not fast enough to measure with dual comp technique. So we switch to another cage compound. So there are other cage compound, like here on the right side. This is a para-hydroxyphenacyl cage group, and this already is cleaved at the excited state after photolysis, and you have this in less than a picosecond GTP formation. Yeah, the disadvantage of this is that you have to synthesize that in your lab. This nitrophenol acylcage group you usually can buy commercially. Yeah, but now we synthesize this para-hydroxyphenacyl GTP and measured this also with dual-column spectroscopy. And indeed, we get now a much faster formation of the GTP. We still see a very fast transient here, but this might be just a heat artifact. But within less than a millisecond, we have the GTP in a very stable baseline here. One additional drawback here was that we don't have an optimal overlap of our excitation laser with the absorbance spectrum of this compound. And also we use thicker samples than we are used to. You know, the QCLs have more power, so you have more energy for the absorbance spectroscopy, and you can use thicker samples, which gives you in principle a better signal. But our laser was then not able to photolyze everything at one go. This is seen here in this figure. We see that we have for about 10 flashes some photolysis and some reaction going on, and only after 15 flashes or so our cage compound was completely photolyzed. So we could do now two things. The one is shown here in C. This is just this very first experiment, so really a single shot experiment. And in D, we have used the mean averages of the first 10 experiments. Yeah, so this gives a better signal to noise. And this is the data I will show later for the proteins as well. So it's a single sample, and we added 10 experiments from the same sample. So these are the cage compound. Now I come to the proteins, the GTPases. So the protein we look most of it is the RAS protein. The RAS protein is a central molecular switch in the signal transduction of the growth signal in the cell. So, yeah, so the growth factor binds to the receptor tyrosine kinase, and then by some adapter proteins, it will be the signal will come to the RAS, whereas the central switch, which then somehow transduces the signal to various downstream pathways, including the one for cell growth. And you can easily imagine why this is important to look at. If this switch is somehow malfunctioning in a way that it's no longer switching off correctly, then signal transduction for cell growth is always on, so the cell will always grow. And this is one of the main factors you find in cancer. Let's enlarge this switch here of the RAS. Yeah, so the switch functioned in a way that with GDP, the diphosphate, the RAS is in the off state, and then upstream, if this growth factor binds, you will, by some adapter protein, bring this so-called source protein to the RAS, which opens the binding pocket, and then the GDP can go out of the binding pocket, and the GTP, the triphosphate, will go into the binding pocket. And this leads to a conformational change of the protein. This is now the on state, and in the on state, the downstream effectors can interact and transduce the signal further on. And then the important part is, of course, the switch off. This is the hydrolysis now of GTP to GDP, and this is what we want to investigate now within our spectrometer. Yeah, so it's a GTP hydrolysis. This is GTP hydrolysis. You see a water molecule attacks nucleophilically at the gamma phosphate, and then the bond between the beta and the gamma here is cleaved. And this we want to understand in detail. So if you have GTP in water, hydrolysis also takes place, but with a half-life time of about 200 days. If the GTP is bound to the RAS protein, then we have a half-life of about 30 minutes. So about factor 10 of 5 faster. And another factor 10 to the power of 5 faster is if you have the GET protein. Yeah, so 30 minutes is fast, but if you really want to switch something in the cell, it's still slow. And together with this GAP protein, you have a half-life of about 50 milliseconds. So that is a really fast reaction. And this is a reaction where we really want to have fast instruments like with a dual comms spectroscopy. Yeah, if you just put our RAS protein in an FTIR spectrometer, what you get as the absorbance spectrum of the whole protein is shown here in the blue spectrum. As you probably know, this is governed by the amide 1 and amide 2 absorptions, the CO stretching mode, and the CN stretch and NH bending mode of the backbones. So these are not absorptions of individual groups of the proteins. These are sum of absorptions of the whole backbone. You can use that, for example, for secondary structure analysis, but you cannot really use it for finding a reaction mechanism. If you want to do that, you really have to do different spectroscopy. And in red, you see has a different spectrum of RAS GTP minus RAS GDP. Yeah, as you see, this difference is much smaller than the overall absorption. But if you blow that up, now the axis here, absorbance, is stretched by a factor of 500. Now you see there's actual information, and there are positive absorptions and negative absorptions. So absorptions which are there in the product state, but not in the starting state, and the other way around. And these are now absorptions of really individual functional groups of the protein. Yeah, if you look at this in the model here, for example, what do you expect in this different spectrum? If you do GTP hydrolysis, a vibration of this oxygen, which bridges the beta and the gamma phosphate, this should be there in the GTP state. It should be gone in the GDP state. Yeah, and if you assign this band, as you should then get in the different spectrum, B minus A, a negative band because it's vanishing during the reaction, and the broad absorption of the other parts of the proteins, they do not change. So they cancel each other out, and they are not seen in this different spectrum. And obviously, you can also do this time resolved and look at this absorption time resolved, and by that, for example, investigate how fast this hydrolysis reaction proceeds. Second possibility for a different band in this spectrum is a change in the environment. If you have for example, a loop in the protein which moves during the reaction, you have another environment, and then you get a shift of the band, or more specifically, if you have, for example, a change in the H bonding of a bond, then you will also get a shift. Yeah, so the loss of an H bond should give you a higher force constant of the bond, and this should give a blue shift of the band. Yeah, so in the different spectrum, you should then see this kind of this double band. Now put these two things together, the caged compound and the protein. So RAS with caged GTP in the binding pocket. Let's do this experiment. So we put the sample in the spectrometer, we get the absorbance spectrum of the whole RAS protein, but then we say we do different spectroscopy. So we say this is just our 0 line, and then we wait a while until everything is stable, and then we measure the reference. We come with the laser flash. After the laser flash, we get a different spectrum of RAS caged GTP and GTP, and then you see that there's a reaction taking place, and this reaction is the hydrolysis reaction where we come from the GTP to the GDP state. This moves on here repeatedly, but in the experiment, you can do this only once, and then you have to change the sample. Yeah, in the end, you have this nice different spectrum of RAS GTP and RAS GDP, so the on and the off state of this switch. Now to learn really something about the reaction, we have to assign these bands to the individual groups. There are two methods. One method is that we do mutagenesis. If you, for example, mutate, here's a 3-ionine 35 group, to alanine, then the side chain absorption should be gone. And if we do now a comparison of the wild type and the mutant protein, you can find the band which is gone and assign this to the respective group. However, this is often too invasive. Often the reaction doesn't really work if you mutate the most interesting groups. This is why we also do isotopic labeling. If you do isotopic labeling by stable heavy isotopes like 13C or 15N, then you increase the mass of the atoms of the vibrations, and by that you have a redshift of the absorption. And this is always non-invasive, and this helps always to assign the bands. Yeah, here's an example. In black, again, the on-off difference spectrum I've shown you before, and in red, the same difference spectrum, but now with the threonine labeled by 13C and 15N. And you see nicely that this band here is now downshifted, and this band can clearly be assigned to the threonine 35. And this also, if you look at the structure, makes sense. Yeah, in this on state, you have this threonine 35, the CO backbone here in the hydrophobic environment. This is why you see it nicely blue shifted compared to the off state, where it's more or less pointing towards the bulk. Yeah, coming back to this picture, I told you that there's also the GAP protein, which further accelerates the reaction. So you can also measure this. Yeah, this was RAS alone. And then together with the GAP protein, you get actually two different intermediates. And now you can try to find the order of events there and see what's happening first in the hydrolysis reaction. And one example I will show you, this is the movement of the so-called arginine finger towards the binding pocket. Yeah, so this is here. What you see is the GTP, which is somehow also directly touched by this arginine finger of the GAP protein, which is a positive charge, which helps this hydrolysis reaction. And by also isotopic labeling, we assign the vibration here and could then also show when this arginine finger moves into the binding pocket and catalyzing this reaction. Yeah, so one methodology is always that we look at order of events, but we can also combine our work with computational models, and then we can compare whether our experimental spectrum coincides with the calculated model. And if this is the case, we know that the calculation is correct, and then we can get very detailed information from this calculation. So for example, you get the geometry with a very high accuracy of about 0.01 angstrom. And now if you look at RAS of the GTP in water, in RAS and in the RAS DAP, you see, for example, that's the difference of the beta and the gamma phosphide fate. So this is where the bond breakage occurs. It's getting longer in the cases where the reaction is faster. So this makes sense. And also another thing you see is that if you look at the partial charges here, which you also get out of the calculation, there's the charge of this phosphor atom, it's getting more positive. And by getting more positive, it's more prone to the nucleophilic attack of the water molecule. So these are two of the key reasons why we have this acceleration of this reaction here. Okay, then I will come to the experiments we did with the dual comps. We didn't use the RAS, we used the heterotrimeric G protein because here the reaction is even faster. So this was more interesting. And as you know, from the name heterotrimeric, so there are alpha, beta, and gamma subunit. And the alpha subunit, that's what's shown here in yellow and in orange. And you see in the middle of these two domains, there is the nucleotide, the GTP again. So it's another GTPase, the G alpha, and it's again a molecular switch. It's important in the cyclic AMP pathway and also connected to a lot of diseases. If there are mutations, you find it in the McCune-Albright syndrome, in various tumors, and the cholera, or also the pertussis toxin, they also interact with the G-alpha protein we see here. Yeah, what we did here before was assigning the vibrations of the GTP, alpha, beta, and gamma phosphate. Yeah, so we knew where is which vibration. And then we looked, for example, at environmental changes. We looked at mutants now of the G-alpha protein. Yeah, here we see the G-alpha again with the nucleotide in the middle. And I highlighted now here the residues of G-alpha, which do hydrogen bonds with the nucleotide. Yes, for example, the lysine 46 or the threonine 48. Yeah, and I will repeat here. If you cleave a hydrogen bond, what you expect is the blue shift of the vibration. And yeah, let's look at the data. So this is now the lysine 46 to alanine mutant, which according to the X-ray structure should sit between the beta and the gamma phosphate. And indeed, we get the blue shift of the gamma phosphate band and of the beta phosphate band. This is also important for hydrolysis. For the catalysis, as you see, this mutant is much slower. Now you can imagine that lysine is positively charged, and it will somehow move charges in the GTP, and by this it's important also for the catalysis. Then we looked at the threonine 48, yeah, which has a hydrogen bond to the alpha phosphate, and now beta and gamma are not changed, but we have a blue shift of the alpha phosphate band at 1243. Yeah, and this is exactly the position where we also had the laser, the dual comp laser for our experiments. So let's look at this vibration. So what we see now is the comparison of this FTIR data with dual comp data. Here, the time resolved data. So this is a reaction takes 29 seconds roughly, and you see now in black the data of the FTIR experiments we did a few years ago, and in red the new results with the dual comp, and you see that we get practically the same rate here. And what you obviously see is the first data point for FTIR here was at about 40 or 50 milliseconds, and with the dual comp we have a lot of data before that, some orders of magnitude. And then the spectral comparison here is also nice. You see the photolysis spectrum, so the difference of the cage GTP and the GTP state. Again, the comparison FTIR versus dual comp showing the same, and then the different spectrum of the hydrolysis here. And again, both show very similar spectra. So we have a lot of room here for faster reactions, so we can add the gut protein here, and this is especially interesting here for the G-alpha, because if you look at X-ray structures, you cannot understand why there is actually a hydrolysis. Yeah, so this is here the overlay of the structure with and without the gut protein, and it's practically the same. So the changes are very small, cannot be resolved by X-ray, but probably they can be resolved by FTIR. And now we can try to measure this very fast reaction with a dual comp spectrometer, and this is done here. Now, this is now the complex of the G-alpha with this so-called RGS4, which is the gut protein of the G-alpha here. And here is now again the hydrolysis reaction. And we see now we have a half-life time of 90 milliseconds. Yeah, I added here this line would be the right of this line would be the first data point for the FTIR, and you see with FTIR you would not be able to resolve this reaction, but here we have very nicely the complete reaction. And even in addition, as you see here in C, we have two more processes, one with 38 microseconds and one with 86 microseconds, which proceed before the hydrolysis reaction, which are again probably some conformational changes or some groups which move towards the binding pocket, which change somehow this alpha phosphate band. So you can nicely get this 3D graph here, where we see now the complete spectral changes from 4 microseconds here up to about 10 seconds, I think is the last data point. And this is done with one single sample of this protein. Yeah, so this is the data we took in February, and yeah, obviously we want to proceed there, doing again isotopic labeling to assign what's going on there on the microsecond timescale, but also since yeah, there are so many more CAGE compounds available, there will be several other proteins we are interested in, which are now possible to investigate by single-shot experiments. Yeah, I would like to acknowledge. So on the left side, these are the people who are on the manuscript we have under review in analytical chemistry under revision, hopefully coming out soon. And on the right side, we have Daniel Mann, who did the FTIR work on the G-alpha, Jonas Schadner, who synthesized the CAGE compounds, and Till Rudak, who did the calculations. And thank you for your attention.
Thanks, Carsten, for this excellent talk. And I invite everybody to drop us questions. You can use just right away the Q&A button underneath. Otherwise, I will just right away kick off the session because one question came in already. You showed really nicely some data on microsecond resolution, Carsten. My question actually, where you compared specifically here, where you compared some data with FTIR, and you mentioned the first point was at 50 or 60.
Yeah. I mean, I shown before in the first slide like 10 milliseconds. In 10 milliseconds, you get an interferogram. Usually, if you have a protein reaction, signal to noise is not good enough to really use a single interferogram for data analysis. So usually we do this, double-sided forward backward mode, and then add this 4 interferograms to 1, and then with 4 wave numbers resolution, I think what we use here, we get 50 milliseconds. But this is for the smaller proteins. If you have larger proteins, we sometimes even add like 10 interferograms or so and have then 100 milliseconds.
Okay. What was the resolution? Just
4 wave numbers.
4 wave numbers. Okay. Mm-hmm. Also with both with FTIR and
we did so for the with the dual comp as you have shown, we have much more data, and we can kind of then decide depending on signal to noise how many data points we want to merge to one point to have a better signal to noise, and we decided to have it similar to the FTIR, so we have a 4 wave numbers resolution there.
Thanks. Then there's another question actually just from Luca Quaroni. Do you have the option of focusing the beam? Probably that's a question for me. Yes, we can do focus the beam. So as you have seen, there are several accessories available. So inside the sample compartment here, you can, so that's actually not the sample compartment here, we just changed the laser, but I can outline that a little bit with another backup slide. Here you see the entire range actually, where you can place several accessories right away in the sample compartment. So you can focus the beam, you can use ATR units, you can use gas cells. So there's actually a lot of possibilities with these first commercially available dual comp spectrometer. So there's another question from Muriel Lepere. Do you think dual comp spectroscopy will replace FTIR in the future in such kind of researches? Probably a question to you, Carsten, because I'm probably a little bit biased.
I mean, it always depends what you want to measure. I mean, the advantage of FTIR is obviously that you have the whole mid-infrared spectrum, but I think in general, I think that QCLs and especially for time resource spectroscopy, the dual comp will be a game changer, and it will yeah, probably replace FTR in at least in many places. So the obvious drawback so far is the spectral range, which is I think 60 or 80 wave numbers now, and it will probably get broader with time, like 200 wave numbers. I think that's something Florian can.
That's right. There's actually always a question, actually, how broad can it go? And this was really nicely covered just by Jérôme Feist. There's always a question, actually, can we go to 300 or 400 wave numbers? So currently, we have already some results on that, up to 100. That was measured last year. And Jérôme mentioned also like 250 is definitely feasible. Currently, all the laser broadness, also which are specified at IRsweep, is 60 wave numbers. And there's actually just another question here about high resolution. So that a little bit depends on specifically when we sweep the comps for high resolution, we need to clarify actually if the laser is really sweepable. So I just quickly actually outlined what kind of lasers we have. So the high resolution spectroscopy, and here I really talk about better than 0.001 wave numbers. They have to be specific characterized. Okay, then probably another question. Why are the larger proteins more difficult to measure in terms of time resolution?
Yeah, this is because of the background. I mean, I've shown you in the one slide the RAS protein, the absorption spectrum, and the small difference spectrum. And if you have the RAS protein, which has, I think, 180 amino acids, yeah, so you look at single groups with the background of 180 amino acids. And then if you have a protein which has twice the number of amino acids, you have twice the absorption of the protein, but still the same small signal of a single side chain. Yeah, so the problem is that you have more background, and if you have more background, you have less signal, or you need yeah, thicker sample, and this gives you less signal. But then again, with the QCL, you can measure this much easier because you can use a larger path length. Yeah, for these experiments, actually, we have had, I think, 60 microseconds, 60 micrometer path lengths. And for the FTR, you usually use 6 or 10 micrometer. So this gives you around a advantage of the factor of about 6 for the signal.
Yeah, I see. Actually, also, is there also probably an advantage to measure through ATR with several reflections with that with dual comp spectroscopy?
Yeah, in principle, I mean, what you could do, you could use then more reflections to get more signal. Yeah.
I think that's one of the big advantages, as you outlined, that you can really go through thicker samples. And yeah, as you said, 60 micrometers we measured already. Depends a little bit on the solvent. You can really go to I don't have now the picture here.
Yeah, sure. But for the proteins, we have to use water, and then
exactly, which is a very strong absorber, right? Okay, let me see if we have some other questions coming up here. Oh, just here quick, actually, we showed those results here with microseconds. What was exactly the fitting model which you were using to get those half times? That's
yeah, so this was a global fit of the complete data set you see here in D also.
Yeah.
And so we had overall 3 three exponential components, and you see nicely that there are two very fast components. We don't know yet what actually is happening there. We know for sure by comparison that this 90 milliseconds is the hydrolysis reaction, and what's happening before, it's probably some movement of amino acids, but that we have to assign again by isotopic labeling or mutagenesis to assign.
I also want to thank from our side also Raphael Horvath, because he did quite a lot of those measurements during just before the lockdown. That was now almost a year ago. And if there are not more questions coming up, I just want to highlight also a next Zoom webinar, which we will do which we will host in April, 29th of April, with Josef Michel, and Markus Geisler will actually host this webinar. It will really nicely show the use of the IRsweep for photophysics experiments with the UC Boulder. And the nice thing is actually also, it's also speed measurements for you see here with microsecond time resolution. And the nice thing is we're using several laser modules. So here you see 1250 data, 6020, 1675, and all together really covers really nicely the range of already 218 wave numbers. Of course, you have to change the module, but like this, you can record quite a lot of data over that area. Then I just got off the phone recently with Cedric. He will also support us, so we will do probably a webinar in May, just to highlight actually what's going on at IRsweep in the future. We did already some nice measurements with Biologic, and we will present that also together probably on a Thursday in May. Then once again, thanks a lot, Carsten. It was a very exciting webinar, and we are definitely looking forward that the paper is published. So it's already submitted, but and it's on bioRxiv if you want to hear more about it. And if you have some additional questions, just shoot us an email or go on our site, www.irsweep.com, for additional applications. Thanks a lot and enjoy your evening.